Hello, everyone. Have you ever pondered how bacteria can rapidly develop resistance to antibiotics or acquire new genetic traits? Today, we will explore the intricate process of bacterial conjugation. This phenomenon is more complex and fascinating than it might initially appear. Let us begin. To start, bacterial conjugation can be likened to a microscopic exchange of genetic information. Instead of exchanging contact information, bacteria exchange genes. This process is a significant mechanism of horizontal gene transfer, allowing bacteria to share genetic material across generations without reproduction. Consider a donor cell possessing a fertility plasmid, also known as the F plasmid. This plasmid is a segment of DNA capable of self-replication. It contains the genetic instructions for constructing the sex pilus, a molecular appendage. The donor cell utilizes this pilus to connect with a recipient cell that lacks the F plasmid. Upon connection, the process of genetic transfer commences. The sex pilus draws the cells together, forming a conjugation bridge. This bridge serves as a conduit for DNA transfer. The donor cell initiates rolling circle replication. It cleaves a single strand of its F plasmid and transfers it through the bridge to the recipient. Concurrently, the donor cell synthesizes a new copy of the F plasmid. As the recipient cell acquires the DNA, it synthesizes a complementary strand to complete the plasmid. Consequently, the recipient cell is transformed into a donor cell, capable of repeating the process. This can be likened to a bacterial relay race with genetic material being passed from one cell to another. The significance of this process cannot be overstated. Bacterial conjugation is a major contributor to genetic diversity. This enables bacteria to rapidly adapt to new environments, which is crucial for survival under adverse conditions. However, this process also facilitates the spread of antibiotic resistance. Certain plasmids, known as R plasmids, carry genes that confer resistance to multiple antibiotics. The dissemination of these plasmids through conjugation poses a significant public health challenge, complicating infection treatment. Conjugation extends beyond basic gene sharing. In some instances, the F plasmid integrates into the bacterial chromosome, forming an HFR strain. This HFR strain can transfer not only the plasmid but also chromosomal genes to the recipient, enhancing genetic diversity. Another variant is F prime conjugation. If the F plasmid excises from the chromosome imprecisely, it may capture chromosomal genes. The transfer of this hybrid plasmid can result in the recipient cell acquiring duplicate genes, leading to novel genetic variations. In summary, bacterial conjugation is a sophisticated mechanism of DNA exchange that drives bacterial evolution and has profound implications for infection control and genetic engineering. A thorough understanding of this process is essential for addressing some of the most pressing challenges in microbiology and medicine. Thank you for your attention. If you found this lecture informative, please share it and subscribe for more in-depth scientific content. Continue to stay curious, and I look forward to our next session.